Thanks, everyone. For those that just joined the recording, my name is Stephanie Hicks. I'm an assistant professor in Johns Hopkins in the Biostats department. And so I am extremely excited and honored to be joining you today to share with you some of my work in the analysis of single-cell RNA sequencing. I decided to focus more of my talk on some methods development, and then I will get to the reproducibility aspects by, for example, initiatives that I'm working in, and then also code sharing and best practices and things like that. I going to start out slow, introducing a little bit of single cell, and then jump into some exciting topics. All right. So for those that are maybe have heard about single cell or sort of like tangentially are aware of it. If you've heard about gene expression data, the way that you most likely have interacted with it is something called microarrays or bulk RNA sequencing. These assays have been around for many, many years now. And what they translate to is for a given gene, we get a molecular readout of the average expression across all the cells in a given tissue. So here we have a brain, for example, we have a tissue sample extracted from the brain. It is made up of thousands or millions of cells, depending on the tissue size. And when we do bulk RNA sequencing, what we really get is just an average expression across all the cells in that tissue for a given feature, for a given gene. You don't actually get to know what the cells are that are present and things like that. An analogy I like to use are Legos, where if you have a big pile of Legos for a given feature, such as the width of each Lego, you get to see an average of widths across all the Legos in the pile. More information than not. So fast forward to 2014, 2015, when single cell RNA sequencing platforms or technologies came online, commercially available. And what that meant is we now get a molecular readout or molecular profile for each individual cell, as opposed to an average across all the cells, which is what bulk RNA-seq does. So the analogy translating it in Legos is, imagine now you get to take your big pile of Legos and you kind of get to organize them into all the different types of Legos. Maybe you get to see the different types of cells in your tissue sample, or maybe you get to see a relative proportion of the fraction of each cell type, for example. Lots of cool things that you get with this data that you couldn't see with bulk. So more formally, single cell RNA sequencing is a process where we take a particular tissue sample, such as a tumor. We isolate and sequence each individual cell. There are a lot of different ways of doing that I'm not going to go into here. But essentially, we get a molecular readout of maybe 20,000 human protein coding genes for each individual cell, where a cell is a single observation. At the end of the experiment, what we get are a set of raw sequencing reads, and we have to figure out how to quantify them, meaning that we want to figure out where the reads map to in a given reference transcriptome or genome. So there on the top right, I'm showing you in the black line a reference genome, and the black box represents a gene that's been annotated in a reference genome. The black lines represent reads that came from the sequencing experiment, and using state-of-the-art algorithms such as like pairwise similarity, we just figure out where those reads map to most accurately in this given a reference genome. And then we count. So we sum up all of those black lines. We say that there were 18 reads that map to gene 1 and cell 1, and we record that in a matrix. We repeat this process for all genes and for all cells. Now, this matrix can be made up of what are called read counts, or maybe they're made up of UMI counts, unique molecular identifiers, for single cell RNA-seq data. The thing you should know about this matrix, though, compared to bulk RNA sequencing, is that this matrix is incredibly sparse, meaning that it has a high fraction of zeros. In bulk RNA sequencing, the fraction of zeros is well under 30% typically, but in single cell RNA sequencing, you could have anywhere between like 30 to 99% zeros, more typically on the high range there. So a lot of zeros compared to bulk RNA sequencing, and that's expected because the cell might not just be needing to express all 20,000 genes at a single time. It's unlikely, in fact, it only may need to be expressing a handful of genes or a couple thousand genes at a given time for its function. So with this matrix, we can do a lot of great things with it. We can make heat maps and plot expression for given genes or for given cells. We can apply machine learning algorithms or just dimensionality reduction techniques, such as principal components analysis, and see how the cells or the observations sit in a low dimensional space, in maybe a linear space or a nonlinear space. Maybe we see the cells begin to separate and cluster in this low dimensional space and do a lot of great things with it. Okay, so the Human Cell Atlas is a worldwide organization with a goal to create a comprehensive reference map or atlas of all cells in the human body. It's a really great goal, and there's been a lot of progress made to date. In 2018, they released a white paper stating that they had a goal of at least 10 billion cells or observations as a first draft in this atlas. That's a lot of data if you begin to think about it. So that's a matrix of, say, 20,000 features, such as human protein coding genes, by 10 billion observations. So that's an incredibly lofty goal, and maybe not very realistic of what current data set sizes are. So let's explore a little bit about some current sequencing or current sizes of data sets. So I picked out 4 papers from 2019 and 2020, and I've presented the titles of those papers. They're very high profile papers in the world of single cell. And I'm showing in red the number of observations or cells that were sequenced as part of each study that you can use for your own analysis if you're curious or wanted to use them. I pulled these 4 papers from a manually curated database. They're shown in the bottom by Valentin Svensson and Lior Pachter. It's a really cool database where you can go explore many different things about, I think there are over like 500 or 1,000 publications where they've manually curated information or metadata about each study. And so anyways, what I'm showing you here is that we are standardly producing data sets of sizes 1 million cells or more. And in fact, these numbers have even grown in 2021 and 2022. So one of the challenges that I'm focused on is trying to think about scalability to algorithms that we use to analyze single cells or single cell genomic data with, but to larger and larger data sets. So for example, I'm going to take a deep dive into one project that I've worked on around unsupervised clustering. And then in the end, or as I pass that point, I'm going to shift gears and talk about other projects in the world of single cell data science. Okay, so I mentioned first I'm going to start out with clustering. So the goal here is to be able to take, for example, 1 million cells and be able to partition it into a set of like K empirical groups using something like k-means or graph-based clustering. So if we're going to cluster 1 million cells or 10 million cells or 10 billion cells, what do we need to do this? First, we're going to want data that can, or we want to work with algorithms that can either work with data in memory, the way we're used to with like a CSV file maybe, or on disk, meaning that these are special file formats such as HDF5, ZAR, TileDB. Those are some examples of them where the data are stored entirely on disk. And by design, we don't load in all the data at once, just simply because it might be a very large data set. It might be 10 billion cells, for example. And so here, our goal is to be able to cluster those full matrices, 20,000 features by 10 million cells, for example. And in that case, maybe the data doesn't even fit into memory, given our computational infrastructure that we have locally or in the cloud, for example. So we want an algorithm, though, that is enabled to work with either in memory or on disk. And these file formats, these on-disk file formats, are the formats that organizations such as the Human Cell Atlas are using. So if you go to explore their data and want to download it, these are the file formats that you will encounter, and then you have to figure out how to work with. We also want these algorithms to be fast, of course. And so here, maybe we want to be able to cluster multiple times, thousands or millions of cells in principal components analysis space or PCA space. Why would we want to do this? So I mentioned, for example, k-means. So k-means is an algorithm that asks a user to provide a parameter, specifically K, or the number of groups that you want to partition your cells or observations in. And so here we want to, for example, run an k-means algorithm with K equals 5, K equals 6, K equals 7, K equals 20, and so forth. And so we want to be able to quickly cluster these data multiple times. And it might be the full matrix, or it might be some reduced matrix where we've taken our 20,000 features and we reduced it down to maybe the top 1,000 PCs, for example. All right, so I've danced around k-means a little bit. So let me dive into k-means formally for those that are not familiar with it. So at a high level, the algorithm takes in a set of n data points, such as 1 million observations or cells, and I call that matrix X, and then a user provided parameter K, where the algorithm k-means partitions the data X into K clusters. Formally, what it's doing is it's minimizing a within cluster sum of squares criterion, where again, X is our data matrix, our 20,000 features by 1 million cells. And then we have mu-I, which represents centroid profiles for a set of K groups. So let's say that there are 5 groups. There's a mu1, mu2, mu3, mu4, mu5, and we want to estimate those parameters for those 5 groups. So in practice, the way that we do that is an iterative algorithm. It's actually called Lloyd's algorithm. There are many different algorithms to be able to do this in practice, but Lloyd's algorithm is like the default one in base R and Python. So it's probably the one that you interact with the most. This algorithm, Lloyd's algorithm, is based on 2 steps that iterates back and forth. One is called an assignment step, and one is called an update step. In the assignment step, what we do is we start with a given set of centroids. So a given set of parameter estimates for mu-I could be just random. And then what we do is we assign each observation to its closest centroid. Once we have a label or an assignment for each observation, then we say, I'm going to take all the observations in group 1, and I'm going to compute a new centroid for all the observations in group 1, putting the centroid at the center. And then I'm going to do the same thing for the other the other groups. And in that way, I get a new updated set of centroid parameter estimates, or mu-I. And then I iterate back and forth between assignment and update. In general, this algorithm works great. It's widely used in all walks of science and mathematics. The problem is, in particular, when you work with data of that's just large. So for example, in our application, when we're talking about working with 1 million cells, for example, you not only have to store all the data in memory, so those features, all those 20,000 features by 1 million cells, you also have to store all the pairwise distances between each observation and its closest centroid. That storage requirement and those calculations, those matrices, just get really big as the data become big. So I looked around for algorithms that would enable scalable clustering for larger and larger data sets. And there are a couple of ways of doing this. I settled on mini batch k-means. I want to explain what it is, and then I'll talk more about it. So mini batch k-means is a really intuitive algorithm that's very similar to k-means, except at each iteration in the algorithm, the idea is to use only a random small subset of the data, and they call that a mini batch. So the size of the subset could be, for example, 1% or 5% of the data. That's an absolute or relative number. Or you could talk about it in an absolute sense. It could be 100 observations or 500 observations. Either one is fine. But the idea is, instead of working with all 1 million observations, you're only working with, say, 1% of the observations at a given time in the algorithm. And so the idea is you load in 1% of the data into memory. you update and estimate parameter estimates, you spit that 1% out, you then upload or input the next 1%, update the parameter estimates and spit that out. And you iterate over the whole data set. But the beauty is you never use the whole data at any given point in the algorithm. And in that way, you're sort of like capping how much data is being processed at one point of time. So in this way, you don't need to store the whole data set into memory. You only need to store a subset of the data set, specifically the mini batch and the k-centroids at any given point. At each iteration, only the distances between the mini batch and the k-centroids need to be computed. So that's a much, much smaller distance matrix. And in this way, mini batch k-means is a really natural candidate for working with on-disk data, such as HDF5 or TileDB or SAR. So there are some existing implementations of mini batch k-means. One is in the R programming language called cluster R. When you go to use it though, the data has to be of a base numeric matrix type. And if you don't know what I mean, just know that these nice on-disk file formats that we want to use don't work. You have to store the data in memory to be able to execute the algorithm. There is another implementation inside of scikit-learn in Python. Here, the input data can be stored in an HDF5 file, which is great. The downside is when you go to execute the function, there's a step at the very beginning that essentially just realizes the whole matrix into memory and then proceeds with the mini batch k-means algorithm. And it really kind of like defeats the purpose because you have to load in all the data set into memory at first. So we set out to implement our own version within the R bioconductor community, and we called it mbkmeans. The input can be any matrix-like object, could be in memory, could be a delayed matrix, an HDF5 matrix, it could be a sparse matrix, and so forth. And the idea here is just for us to be able to provide a package that enables scalable versions of k-means. And so if you are working with k-means in any form, you could just simply replace k-means with mbkmeans, and you're off and running in terms of a scalable version of k-means. Then I just want to mention that this package was developed for single cell analysis, but it's a general algorithm that you can use in any application that you're using k-means, like computer science for classifying images, or like for clustering images. Anywhere you're using that, you can use mini batch k-means. So let me tell you a little bit about it. The first thing I want you to know is that mini batch k-means is very memory efficient and very fast. So what we did is we started with a data set of 1.3 million neurons, mouse neurons from the brain, and we downsampled to smaller data set sizes of 75,000 up to 1 million. So that's on the x-axis there. These are different data set sizes that we downsampled from a larger matrix. And on the y-axis, I'm going to compare the max memory used on figure A on the left and the elapsed time on the right from running 3 different clustering algorithms. So k-means is shown in red. Mini batch k-means in memory, meaning that we read all the data into memory and then we perform the mini batch k-means clustering algorithm. And then in blue is mini batch k-means with a data set that has data stored entirely on disk in an HDF5 file. So what you see is k-means is very memory inefficient and can get slow very quickly if you're working with data sets more than a couple 100,000. Well, if you just simply replace, if you don't even want to deal with an HDF5 file or SAR or TileDB file, you're like, I can store my data in memory, great. You can get huge cost savings in both memory usage and in time or speed if you just simply replace k-means with mini batch k-means. And that's huge. That's like basically free memory that you're saving and like saving a lot of time. Now, if you are interested or you work with data stored on disk, such as an HDF5 file, you can cluster 1 million cells in less than 2 gigabytes of memory in under 10 minutes. which is like phenomenal. And the reason is because we're thresholding the amount of data being read into memory and those pairwise distances being calculated at a given point in time. So if I'm only reading in 1% of data, or like 100 observations at a time, basically that will be capped. The memory usage is capped because it will never really get larger than those 100 observations, or the memory required to store those 100 observations in those pairwise distances at a given point in time. So that blue line there that you see in the bottom left, where it's flat, it will just continue to stay flat as it gets larger, as the data becomes larger and larger, which is really cool. You also may be curious about accuracy. So whenever we talk about cost savings in terms of speed or memory usage, there's usually like a catch. The catch is, or the take-home message here is that mini-batch k-means is just as accurate as k-means for a large enough batch size. So I've sort of like danced around this concept and introduced the batch size to you. This is a user-defined parameter in addition to the choice of k, where the batch size essentially is how much of the data are we using at any given point in time for the algorithm. So here I'm showing you 3 different data set sizes, starting on the left with 5,000 observations, in the middle with 10,000 observations, and on the right with 25,000 observations. On the x-axis here, I'm now showing you absolute batch sizes. So I'm working with, for example, 100 observations, 500 observations, 750 observations, or 1,000 observations while running, for example, mini batch k-means. And on the y-axis, I'm showing you the within cluster sum of squares criterion. This is essentially the same as an elbow plot that you might have seen if you've used k-means before to help you determine the right k. It's a way to help us assess the accuracy using an internal validity metric, for example. And so what you see is that k-means is flat as a function of batch size, as it should be. There is no batch parameter for k-means. But mini batch k-means in both green and blue, which are essentially the same in terms of, they are the same in terms of accuracy. What happens is for small batch sizes, you basically get noisier estimates of the centroids. And as you get a larger and larger batch size, what happens is the parameter estimates that you get from mini batch k-means and k-means converge, and you get comparable within cluster sum of squares criterion. So the take-home message again is for a large enough batch size, such as 500 observations, you basically get the same results or the same accuracy as k-means, which is really great because working with 500 observations or even 1,000 observations at any given point in time is huge cost savings, and working with 500,000 observations like k-means. So you get those huge cost savings in terms of memory and speed, along with not so much of a loss in accuracy. We then applied this to the full 1.3 million cells. I'm showing you a UMAP representation of the 1.3 million cells colored in a hex bin plot. It's a great way to plot a lot of data that avoids overplotting. The package is called scHex. It's available in Bioconnector. And I've colored each one of the hex bins by the majority of the observations for each predicted label, each predicted cluster label from mini batch k-means. So there's a cluster in red, there's a cluster in blue, a couple clusters in purple, and so forth. So what did we do to get this? We took our full 1.3 million cells, we applied PCA, and we calculated the top 50 principal components. And then we applied mini batch k-means to the 50 by 1.3 million cells. And we clustered 16 times, meaning that we tried out k equals 5, k equals 6, k equals 7, and so forth. That whole clustering process took 3 minutes for the full 1.3 million cells. In contrast, we compared that to state-of-the-art in graph-based clustering, which is available, for example, in Bioconductor or Python or ScanPy. So in Bioconductor, the state-of-the-art is a shared nearest neighbor. It's to first calculate a shared nearest neighbor graph on the top 50 PCs, followed by Louvain clustering. And in ScanPy, the state-of-the-art, at least as the time of the publication, it was BBKN or batch corrected KNN followed by Leiden clustering. And those two processes took 35 minutes and 48 minutes respectively, thereby showing really cost savings in terms of speed for mini batch. And I will say, if you check out the paper, we did go on to compare the clusters that we got from graph-based clustering to mini batch k-means, and they're really comparable, and they're available in the supplement. If you're interested in learning more about the methodology or the package, I encourage you to check out the paper that's available in PLOS Computational Biology. All right, so next I'm going to switch into a project for dimensionality reduction with single cell data. Are there questions, or should we hold off until the end?
Hey, Stephanie, actually there is a question. I think one question is asking about in the previous brain, the UMAP data that you showed, how long that does your software take to plot that? The question asks PCA, but I think it's talking about the UMAP of the brain, 1.3 million cells. How long does it take for you to plot that?
How long does it take to plot PCA, or to calculate PCA, or to plot the UMAP?
I think the question is how long does it take to compute the principal components?
That's the longest part. That is a very insightful question. So this is like this is like the holdup for the slowest part of single cell analysis, is to calculate the principal components. There are approximate or randomized versions of PCA that you can use to like speed up this process. And even that is not so fast. So yeah, I think if more research could be done on speeding up the process for PCA or dimensionality reduction techniques that preserve distances between observations in a very fast way, that would be huge. There's another question. Should I keep reading, or what would you prefer?
So the other question is with the batch size increasing. I think we can just address this question, and depends on how many slides you have left. So with the batch size increasing, the accuracy usually decreases. But in your case, is it opposite? And why is that being opposite, if that's true?
So I don't know if I understand the question, but maybe I go back here. A within cluster sum of squares criterion that's smaller is better. In this case, like you would want the within cluster sum of squares criterion to essentially like converge to whatever it is to k-means. And when it's larger than k-means, it's noisier than k-means, meaning that it's less accurate, or essentially like, so within cluster sum of squares is doing is it's basically saying how tight are those clusters. And so whenever you have a batch size that's really small, those clusters are not really tight. And then as you increase the batch size, essentially like the cluster density essentially gets tighter. And so it converges, like the criterion converges to what it is for k-means. It like stabilizes is a way of thinking about it. Would you like me to keep reading? I'm okay either way.
Okay, I think while we're on the topic, so for different data, do we need to tune batch size?
Yeah, great question. So in our hands, a batch size of around 5% works really well for most data sets. I will say that there are caveats to that though. So like if you have a data set that had a very, very rare cell type or a very rare observation that or a class of observations that you're interested in estimating or like capturing, the batch size is going to play a huge role in the sense that you're loading in 5% of the data at a given time to be able to estimate those centroid profiles. And if you're not getting like a sampling of those rare populations that you're after, mini batch k-means might not be the right choice for you. But in general, unless you have an extremely rare population, k-means and mini batch k-means is going to work really well. So it might be the case that you need to tune the batch size and you have to try out different ones. But in our hands, in general, the batch size, as long as it's above like around 500 or 1,000 observations for like these sizes, it's basically the story is the same as you get larger and larger data sets. Then it works really well. But just again, saying the caveat, unless there are examples in which k-means and mini batch k-means might not work well for your setting. Okay.
Hey, thank you, Stephanie. Let's keep going.
Okay, sounds good. Okay, so in this next section, I'm going to talk about dimensionality reduction for single cell data. So I'm going to begin by motivating what we do just in like what's standard to do dimensionality reduction with single cell. So the standard approach is to essentially apply the tools and the techniques that we developed for bulk RNA-seq and just apply them to single cell. And I'm going to show you why that's a bad idea. So when we do this for bulk RNA sequencing, and when we do this in application of single cell RNA sequencing, what we do typically is to apply some kind of transformation to the data, and then, or normalize it and then apply some kind of transformation, and then apply the dimensionality reduction technique, such as principal components analysis. And the reason that we do this is because methods such as PCA, or principal components analysis, were designed for a particular type of data, namely normal looking data, Gaussian looking data. The problem is with single cell, data are not very Gaussian. First of all, they're counts. They're a count of 1, 2, 3, 4, and so forth. And there are a lot of zeros, as I mentioned at the beginning of the talk. So in bulk RNA sequencing, this was less of an issue. There were not so many zeros. It was still counts. And so what people did was they essentially transformed the counts to make it look Gaussian and then apply PCA. So for single cell, I'm going to show you what happens when you do this. I'm going to give you an example where this is a data set of monocytes. It's a data set that I'm going to call a set of biological replicates or negative control data, where we just do not expect a lot of variation between these cells. These are mostly homogeneous. And so I'm showing you the distribution of expression for one gene in that data set. So I'm showing you the distribution of expression across all the cells in this monocyte. Next, what we do is we take this distribution of expression shown in the left, and the first thing that we do when we apply these standard transformations to be able to put it into PCA is to normalize it. So what we do is we take the counts for a given cell, we sum them up, and for every count in a given cell, we divide it by the total number of UMI, and then we multiply it by 1,000,000. That's called counts per million. And so the transformation to the figure on the left to the figure in the middle is the application of counts per million. It's a standard normalization that we do in single cell that again was developed for bulk RNA-seq. And what you see is all the counts that are not zero sort of get shifted over to the right a little bit, and all the counts that are zero remain zero. After we apply the counts per million transformation, the next step is to apply a log transformation. And the reason we do this is a form of variance stabilization. I'm not going to go into those details, but that's why we apply the log transformation. Also, it makes the data look a little bit more Gaussian. So what you can see going from the middle to the right is that the blob of points that are non-zero sort of get shifted over even further to the right. And if you squint, it kind of looks Gaussian. I know there's like two modes, just ignore that. But in general, it kind of looks Gaussian. And then again, you have this point mass, or this at zero, that remains zero. And there's a pseudo count in here. Sorry, I should have mentioned that. It's the log of the CPM plus a pseudo count of 1. Okay, so what happens when you apply PCA to these log 2 transforms counts per million single cell RNA-seq data? So here's the PCA, or the first two principal components of those sets of monocyte, those monocyte cells. Every point here is a cell. And in this data set, because it's a negative control data set, or a set of biological replicates, we would expect a blob. We would expect essentially no structure in the data. And what we see is sort of like these elongated shapes where the cells in the top right are colored in blue and the cells in the bottom left are colored in red. The color represents the fraction of zeros for a given cell. Essentially what's happening is it's calculating this spike at 0. So this spike at 0 is for one gene, but essentially what's happening is the cells that happen to have more zeros in it get shoved up on the top, on the bottom left, and the ones that have less zeros get shoved up on the top right. This is technical variation that we would not want to find. If somebody applied PCA to this and then did unsupervised clustering, they might suspect that there's 2 clusters of cells. And in reality, what you're discovering is just differences due to the number of zeros. And so this motivated us to create a tool to generalize PCA to, instead of data that were designed for Gaussian data, data that are designed for counts. And we started out by trying to figure out what is the right count model to use to be able to generalize PCA. So what we did was we took the set of monocytes that we had and we calculated the mean expression along the x-axis and the fraction of zeros along the y-axis. And we fit 3 different models to this data: multinomial, Poisson, and negative binomial. And you can see that the observed variation that we see in the data, the black spots, corresponds very closely to these 3 different types of models, sort kind of like equally well. And this is great because in the setting where we expect not a lot of biological variation, this enables us to determine that, yes, we can use a multinomial model or Poisson model or negative binomial model to be able to model these data in a very accurate way. So what we did was we essentially generalized PCA for non-Gaussian data. And the idea was to take the PCA objective function, which is formally the minimization between the data Zij and some low rank representation Ui, which are called factors or principal components, and Bj, which are often called loadings, in some low dimensional space RL. We're interested in estimating U and V as like PCA. That's the PCA objective function. And Zi is typically normalized and log transformed, like I showed you before. So that PCA objective function, the minimization, is equivalent to maximizing the likelihood under a Gaussian model. This is a well-known fact. So again, you're just trying to estimate those parameters U and V there. And our idea was essentially to replace the Gaussian model with a Poisson model and a slightly different parameterization with a GLM model, where Ni represents the total number of counts for cell I, and we're again interested in estimating Ui and Vj. So that's what we did. We formally developed an optimization algorithm to estimate Ui and Vj. That method is called glm-pca. Because I'm running low on time, I'm going to skip over the details, but you should know that the full algorithm itself is a non-convex optimization. It's prone to local optima and kind of like numerical divergences, and it can be really slow computationally. So while this is great and it worked really well in practice, it was very slow. So we implemented a kind of like an approximate version of glm-pca. And the idea was, we know that PCA is like beloved by biologists. They don't want to install typically fancy packages and wait for something to run. And if we can get the data as close as possible to normalize, quote unquote, normalize, so we can use PCA, that's going to have a lot of impact with practitioners who are going to use these tools to analyze their own data. So we decided to kind of like PCA is the goal. And we know that this is a kind of a linear Gaussian model. Another way of thinking about transforming the data is to make the errors or the residuals look Gaussian. And what I mean by that is we know that the model, for example, a Poisson model, fits very well to the data. I showed you that in a previous slide. So for a given gene, we estimate the parameters. We say, okay, in the null setting where we don't expect any biological variation, we're going to expect a set of counts under a Poisson model. And so what we say is we're going to fit the parameter, the mean value, or the expectation of the Poisson model, so lambda or mu, for example. And so we can calculate what we expect under a Poisson model and what we observe for a given gene. And we calculate that difference. And if that difference is small, then we say there's not a lot of like variation compared to what we expect under a Poisson model. But if that difference is big between what we expect and what we observe, we say there's more variation than what we expect under a Poisson model, or there's like a bigger difference than what we expect under a Poisson model. And that's an interesting gene. So what happens is we fit these null models for every gene, these noise only models, and then we can apply PCA, because if you're familiar with generalized linear model theory, the residuals that are called formally deviance residuals or Pearson residuals under these models, those residuals are Gaussian. And so that's what I mean by transforming the data such that the errors or the residuals look Gaussian. So we calculate these residuals, and then we can pipe that straight into like vanilla PCA. So we call that approximate GLM-pca. And when we do that, I'm going to go back to the negative control data that I showed you before. So this is the plot of PCA using counts per million and log2 transformed data. This is what happens when you apply GLM-pca in our negative control data. We no longer see like the weird sort of elongated structures that we found before. So all of this work is available in a paper in genome biology led by Will Towns. There is a package that's available on CRAN, and it's called glm-pca. This is a package that can be used for principal components analysis for any kind of count data. It's, again, was motivated by the use of single cell, but that's why it's in CRAN and not in Bioconnector, because it was designed really for anybody who wants to do dimensionality reduction with counts. And then there is a bioconductor implementation as well called scRNA. scRNA has no meaning. It's Will Towns just kind of like made it up, I think, to mess with me and all the authors. And it's essentially a wrapper around GLM-pca, but it kind of comes with all the bells and whistles that one might want if you're working within the bioconductor community. And so I am running low on time, so I was going to do a demo. I'm going to just kind of skip over these, if you can hang with me for just a minute. And then I have like one more very small section, and then I'll stop. So the last challenge that I work in is education. So I'm in a school of public health, and I'm very passionate about wanting to help train the next generation of researchers. So I set out to write essentially an online book of how to bring in or integrate many different packages within the bioconductor community to be able to analyze data in a really coherent way. And in 2018, there were over 100 single-cell RNA-seq packages or methods and packages to be able to analyze single-cell RNA-seq within Bioconnector. So I wrote a paper to help sort of organize the thoughts and an overview of single-cell analysis within R and Bioconnector. And we talk about ways to standardize and store your data. We talk about workflows that you can use, starting with QC and quantification, all the way through normalization and feature selection and dimensionality reduction, clustering, differential expression, trajectory analysis, and so forth. But the real beauty and the thing I'm most proud of is this online book. It's written in book down, and it's built on the Bioconductor build system, I think every 2 weeks nowadays. And so the idea is it's a set of 42 chapters, including 18 complete end-to-end workflows of different types of data. So site-seq data, 10x data, smart-seq data, all sorts of single-cell data that you can use to analyze your own data. There are lots of philosophical discussions about single-cell analyses. So one of them that I love is I often get this question from collaborators, what are the true number of clusters or cell types in my data? And the answer is that's unlikely to be something I'm going to be able to tell you. Clustering will always give you an answer, a set of clusters, and it's all about resolution in terms of wanting to know what you want to associate with some kind of biological idea. And then, of course, there's lots of code for you to be able to copy and paste yourself with example data sets that you can execute and then change out the name of the data for your own data. Yeah, and I want to end by saying I'm also very, very passionate about reproducible research. So outside of single cell analysis, I am actively writing about this. So I've developed some reproducibility standards for machine learning in the life sciences. I've written review articles for reproducible research on public health. And I'm also an editor for reproducibility at the Journal of the American Statistical Association. And I'm happy to talk about any and all of this if you're interested. And I want to stop there to make sure there's time for questions. Thank you, everybody, for your attention.
Thank you, Stephanie. This is a great talk, really educative, informative. I'm going to start with throwing a question, very technical. So you talk about the glm-pca. I think it's a great method. Could you very briefly comment on how compatible or how it will be able to deal with single cell data with strong batch effects? So if there are there ways for us to model the residuals or take them out, or how does that compare to our traditional way doing PCA with strong batch effects, stuff like that?
Yep. So as part of the glm-pca algorithm itself, you can incorporate a covariate that's associated with a batch effect. But outside of that, like essentially, it's designed to work exactly the same way that you would interact with it for PCA. So PCA itself is not designed to correct for batch effects. We do batch correction in other ways. And so depending on like what you're wanting to do with it, you can basically simply replace the components that you get from glm-pca, that you get from PCA with glm-pca. And you should be able to do other things with them downstream, like pipe them into UMAP, for example, or do unsupervised analyses and so forth, if that makes sense.
Yeah. That's good. One of the audience asked, the idea of glm-pca sounds a little bit like SC transform plus PCA. Do you want to explain the difference?
Yeah, so I'm not an author of SC transform, so caveat there, like you should, read the SC transform paper versus like take it from me. But my understanding is that it's a similar idea. And in fact, when we went to preprint the paper itself, there were conversations with the authors of SC Transform at the time, and we kind of came up with very similar ideas at the same time. And so I would say that they are similar, but they're like different choices and like how the parameter estimation is goes forward. And but it's a very similar concept of like wanting to work with residuals under kind of like a noise model, under a negative binomial model, for example.
Sounds good. I actually also have a computational time question. So usually people hate me asking this type of question. So like you mentioned, the single cell data sets are growing bigger and bigger. I'm recently, our research team is dealing with like a data sets. It's called tabular mirrors, which is like 500,000 cells being processed with different types of cell types from 20 different types of organ tissues from mouse. Could you comment on like on several 500,000 cell, that kind of sample size, how glm-pca could potentially run? Are there any suggestions, to boost up the efficiency and accuracy without burning up the CPUs and MEM too much?
So glm-pca will be slow, like full glm-pca in the way that it's currently implemented. So that's why, like in practice, I just use approximate glm-pca. It's in fact like very similar in terms of results. It's less accurate than I would say glm-pca is, but by design. And then if you want to know like what I mean by less accurate or like how less accurate, I strongly encourage you to just go read the paper itself on genome biology. But I in practice use glm-pca. And so in my demo, I was going to show you how I do it. And I'm happy to like send a link if it's helpful. But I created essentially, maybe I'll just link to the website.
I think we still maybe have like 5 minutes. Why not just go through the demo very quickly?
Oh, okay, sure. Let me go there. Okay. So the demo is available on stephaниehicks.com/biocdemo. It's there at the bottom if you're interested in following along. The idea is I'm going to start out with the data set with 4,000 cells only for the purposes of demonstration. And so the data itself is available in a bioconductor experiment hub package. It's a set of PBMC cells, but this entire workflow can be replaced with 1 million cells and will work. So the idea is I'm loading in what's called a single cell experiment object, which is an object that we developed to be able to store single cell data in a really modular and interoperable way. And inside of it, you see something called counts assay. It's a matrix of counts that I showed you with 33,000 genes along the rows and around 4,300 cells along the columns for cells. So you can go investigate this object. You can look at it. For example, you can ask for the counts matrix of this single cell experiment object. And what you get back is something that kind of represents, like kind of feels like a matrix. So it says matrix there at the top, and you see genes along the rows, and you see 1 to 4,300 cells along the columns. Now, the thing I want to highlight is that this is called, it's a type of matrix called a delayed matrix. And if you go to explore what is a delayed matrix in Bioconductor, essentially it's an HDF5 file. So the matrix that I'm showing you right here, while it feels like a matrix, like it feels like I've loaded in and I'm showing you like the first 5 rows of the genes and the first 5 and the last 5 columns of this matrix, it's not. All the data are stored entirely on disk, and I'm only showing you kind of like a trick of like it feels and it works like a matrix, but it's not really a matrix that's read into memory. That's the trick behind this. So that's why you can simply replace everything that I'm showing you with 1 million cells and it'll work. So there are some steps not shown here that are shown in the tutorial. I do remove some low quality cells. I do remove some lowly expressed genes. And I did apply a normalization to do clustering from mini batch k-means. This is only designed to do scRNA normalization, which is necessary to do like standard PCA. So that's all there. Okay. So the first thing I'll show you is what we do with the standard technique. So this is PCA applied to the normalized data and followed by log2 transformed. And so I am showing you a function called run PCA in the scatter package. And what you see are the low dimensional representations of these 4,000 cells. And you see these elongated shapes, like I showed you before in the null residuals, the null negative control data. That's like essentially what's being driven here. If you plot those cells, if you plot PC1 against like the fraction of zeros, you'll find a strong correlation. And if you want to know more, you can go to the 2 publications that I talked about. So next, I'm going to switch gears and I'm going to show you what happens with glm-pca. So I do a couple of things. So this is an alternative to PCA on those normalized data. So the first step is I calculate a set of residuals. So I take our just counts matrix, and for every gene, I fit a model. In this case, I think the default is like a Poisson model, and I calculate a set of deviance residuals. So I have a big error matrix at the end of this. And then from there, I take that set of residuals. You can see the expression values equals binomial deviance residuals, and that's what gets piped in to PCA. And so PCA, the combination of null residuals and PCA is what I use in practice to be able to do quote unquote glm-pca or approximate glm-pca. And that's what I'm showing you along components 1 and 2. It's the cells plot in the glm-pca components 1 and 2 space. And you no longer see these elongated shapes that you saw for PCA. And then we combine that with clustering. So I'm no longer going to show you anything about PCA. I'm only going to show you the glm-pca results. You can do graph-based clustering, or you can also do mini batch k-means clustering. So here I'm showing you, I'm taking in the glm-pca components. That's what reduced method equals glm-pca is. It's saying extract out the glm-pca components and then give that to mini batch k-means, do clustering on mini batch k-means from k equals 5 to k equals to 20. And then we get a set of clusters from mini batch k-means with the glm-pca components. So if you want to like do this in your own data, this is how I do this in practice. And I'll stop.
Great. Great. Thank you, Stephanie. This is super cool. For everybody who want to follow up on this, definitely check out the links I shared within the chat box and the papers that Stephanie published on relating to the methods. I do have one last question. I know we're almost running out of time. I love philosophy questions. So I know we briefly talked about bronze, silver, and gold standard for reproducibility. So one of the focus is asking, what is your take on the minimal requirements for a single cell analysis to be reproducible?
Oh, wow. So there are many aspects to that. I think ideally, I would want the data to be made publicly available. I would want the code that was used to create either the figures in the manuscript or to reproduce the analyses to be put available, like to be shared in some kind of open way, and also to be versioned. So if the data changes over time or if the code changes over time, to be versioned. Yeah, I would say like that's the minimal thing, and some kind of like readme or documentation to maybe like explain and walk a reader through, like if you have a set of scripts that are sort of like jumbled, then just to be able to explain that to them, like what's happening in each one.
Sounds good to me. Thank you, Stephanie. I'm gonna close right now. It's almost 1 p.m. I really appreciate you coming in here today. I also I'm very thankful for all of the 100 people who joined online with me today. And we're going to post this video recording to our YouTube channel. So many of the good stuff that you missed during the talk, and you can always review them back later. Thank you, Stephanie, for coming over. Thank you, everybody. I
Take care,
hope to
everyone.
see you guys in the future. Okay, you guys
Bye.
have a great day. Bye-bye.